Hello everyone. Today we are here to discuss the topic aggressive periodontitis. So first let us know the history of aggressive periodontitis. In 1923, Gottlieb was the scientist who coined the term diffuse alveolar atrophy of the bone. Diffuse alveolar atrophy. During an attack of influenza, he examined the oral cavities of the individuals and found out that in some patients there was severe attachment loss and bone loss. When these teeth were extracted and examined, it was found that various cemental defects were present in these individuals. So, he coined the term deep cementopathia in 1928. In 1938, Wannenmacher was the scientist who coined the term marginalis progressiva periodontitis. Wannenmacher in 1938 coined the term marginalis progressiva periodontitis. He was the one, progressive periodontitis. He was the one who showed the involvement of first molar into the individuals affected by aggressive periodontitis. Now, in 1967 and 1969, Butler and Chaput coined the term localized aggressive periodontitis. Chaput and Butler coined the term juvenile aggressive periodontitis. Why juvenile aggressive periodontitis? Because it affects the individuals of younger age group, individuals who are lesser than 30 years of age. So, the term juvenile aggressive periodontitis came into existence. In 1993, Rani coined the term early onset periodontitis. Early onset periodontitis because of the age group of the individuals affected by this disease. In 1999, American Academy of Periodontology coined the term aggressive periodontitis because of the aggressiveness of the disease with which it affects the individual's bone and tissues. So, what is aggressive periodontitis? Aggressive periodontitis is the disease of the periodontium occurring in otherwise healthy individuals, and it is characterized by rapid loss of alveolar bone in individuals, rapid loss of alveolar bone which affects more than one tooth in a permanent dentition. In this, there is severe alveolar bone loss destruction present, and these bone loss which is seen is that it does not commensurate with the local irritants which is present into the individuals. How is it different from chronic periodontitis? Chronic periodontitis does not affect only single teeth, it affects the whole dentition of the individuals, and there is severe attachment loss and bone loss and severe inflammation present in the individuals affected with chronic periodontitis. It is most frequent form of periodontitis occurring in the individuals, and this is associated with the presence of plaque. Individuals which are affected by chronic periodontitis have severe local deposits present such as plaque and calculus in them. Aggressive periodontitis is divided into two forms: localized aggressive periodontitis and generalized aggressive periodontitis. In this localized aggressive periodontitis, less than 30% of the teeth are involved. In generalized aggressive periodontitis, more than 30% of the teeth are involved. In localized aggressive periodontitis, at least two permanent teeth should be involved, and one of which should be the molar. In generalized aggressive periodontitis, three permanent teeth should be involved other than molar or incisors. Other than molars or incisors. In patients affected with localized aggressive periodontitis, it is seen that there is a robust antibody response. What is robust? Robust is a strong antibody response against the antigens present into the body. So, in localized aggressive periodontitis, robust antibody response is seen. robust antibody response is seen. While in patients affected with generalized aggressive periodontitis, a poor serum antibody response is seen to the antigens. Poor serum antibody response is seen towards the antigens. This is the basic difference between the localized aggressive periodontitis and generalized aggressive periodontitis. Thus, generalized aggressive periodontitis affects the individuals of 30 years of age up to the 30 years of age, and in this the attachment loss is seen in teeth, at least 3 teeth other than first molars and incisors. It has pronounced episodic nature of periodontal destruction, and a poor serum antibody response is seen in the patients affected with generalized aggressive periodontitis. In localized aggressive periodontitis, it is seen that it begins at a circumpubertal age, means it affects the individuals of adolescent age group, and the attachment loss is seen on at least 2 permanent teeth, one of which should be the first molar. And in these individuals, a robust serum antibody response is seen. Now let us one by one know about localized aggressive periodontitis and generalized aggressive periodontitis. In localized aggressive periodontitis, as we have all seen, less than 30% of the teeth are involved. This is a disease of periodontium occurring in an otherwise healthy individual, and it affects the patient of adolescent age group, means the patients of circumpubertal age, and in this the interproximal bone loss is seen on at least 2 permanent teeth, one of which should be the first molar, and not involving more than 2 teeth other than first molar and incisors. It is seen that in localized aggressive periodontitis, the amount of tissue destruction and bone destruction seen is not consistent with the amount of local factors present, like plaque and calculus. Thus, the severity of destruction is not consistent with the amount of local factors present. Now, let us know about the etiology of aggressive periodontitis. For the aggressive periodontitis to manifest, at least 2 requirements are present. There must be a highly virulent organism present. Highly virulent organism must be present. Highly virulent microorganism must be present to cause the disease, and also the susceptibility of the individual. The individuals should be susceptible for destruction of the bone and the soft tissue. Susceptible individuals. The bacteria which is mainly responsible for causing localized aggressive periodontitis is Aggregatibacter actinomycetum comitans, which was previously known as Actinobacillus actinomycetum comitans. Now, this bacteria possesses various virulent factors which is responsible for causing localized destruction. The virulent factors present in AA are leukotoxin. This leukotoxin is responsible for the destruction of macrophages and neutrophils present into the host. Destruction of macrophages and neutrophils. The second virulent factor which is responsible for causing the destruction is bacteriocin. This bacteriocin inhibits the beneficial species into the host and thus causes increase into the colonies of Aggregatibacter actinomycetum comitans. Bacteriocin inhibits the beneficial species into the host. The third virulent factor is collagenase. As we all know, collagenase is the enzyme which is responsible for degrading the collagen. Thus, the collagenase produced by the AA is responsible for the destruction of the soft tissues into the host. The fourth virulent factor is immunosuppressive factor. This immunosuppressive factor is responsible for inhibiting IgG, immunoglobulin G, and IgM into the host, and thus when these immunoglobulins are inhibited, they cannot cause phagocytosis of the aggregative actor actinomycetum comitans. The fifth virulent factor which is responsible for the destruction is chemotactic inhibiting factor. This chemotactic inhibiting factor is responsible to inhibit the chemotaxis of neutrophils. The neutrophils are the primary line of defense which come at the site of inflammation and are responsible for the degradation and destruction of the bacteria. But if these neutrophils are not coming to the site of inflammation, then the bacteria are not destroyed and thus they colonize and increase the destruction. Thus, these are the virulent factors in AA which are responsible for the localized destruction of the tissues into the susceptible host. The second etiology is the immunological etiology. Immunological means the immune response of the host which is responsible for the destruction of the, there are defects into the immune response of the host against AA. These immunological defects are defective chemotaxis of the PMNS. There is defect into the neutrophils and thus they cannot come at the site of inflammation and thus they cannot destroy the bacteria. The second defect is the hyperresponsive monocytes. These hyperresponsive monocytes are responsible for increased production of interleukins such as interleukin 1 alpha, interleukin 1 beta and various other cytokines such as prostaglandins, and these are responsible for the destruction of the bone. The third defect which is present into the immunology of the individual are human leukocyte antigen. Human leukocyte antigen A9 and B15 are the candidate markers in aggressive periodontitis patients. This is very important question of NEET. This is most frequently asked, which are the HLA complexes which are responsible for the immunological response of the host towards the AA. Thus, this is the etiology. Now, let us go to the next slide. Now, the genetic etiology. It is seen that localized aggressive periodontitis run into the families of the affected individuals through various segregation and linkage analysis. It was found in linkage analysis studies, it was found that there is a specific gene located on chromosome number 4 which is responsible for the defects into the neutrophils and defect into the chemotaxis. Thus, aggregative actor actinomycetum comitans colonizes the affected sites. The transmission occurs through autosomal dominant mode of inheritance. The environmental factor which is responsible for causing aggressive periodontitis is smoking. It is frequently seen that the affected individuals have a history of smoking. The current concept is the aggressive periodontitis is a multifactorial disease. Multifactorial disease. Why multifactorial? Because not only a single factor is responsible for causing the aggressive periodontitis. It is seen that accumulation of various factors such as the genetic defect, environmental factors such as stress, smoking have led to the development of aggressive periodontitis. Thus, it is found that aggressive periodontitis is a multifactorial disease developing as a result of complex interactions between specific host genes and environmental factors such as stress and smoking. Interaction between this disease process like cigarette smoking and genetically controlled modifying factors are responsible for the disease manifestation. Now, let us go to the clinical features of localized aggressive periodontitis. It is seen that it affects less than 30% of the dentition in an individual. It is seen mostly into the circumpubertal age in the patients of adolescent age group. The sites affected mostly involved are molars and incisors. Molars and incisors. It is seen that there is diastema present in the upper maxillary incisors. Distolabial migration of incisors, upper incisors is seen. The affected tooth are supraerupted. There is sensitivity present into the affected sites. there are very less deposits of plaque and calculus presence, less deposits present at the affected sites, and these deposits are not consistent with the amount of attachment loss and bone loss present. So, deep periodontal pockets are present. Pockets are present in the affected individuals. Severe amount of bone destruction seen. The patient complains of dull radiating pain at the affected site. Dull radiating pain. During the radiographic examination, it is seen that there is a mirror-shaped, arc-shaped bone loss present onto the affected sites. See, these are the molars, and in this the amount of bone loss present is like this. Such arc-shaped bone loss are present into the radiographic features, and this similar lesions are present onto the opposite side of the arch. So, it is called mirror image. Mirror image destruction is seen on both the sides during the radiographic examination of the individuals. Also, it is seen that there is a burnout phenomena. This burnout phenomena was the term coined by Rane. Initially, it was thought that localized aggressive periodontitis proceeds to generalized aggressive periodontitis, but during the various studies and various examination, it was noted that localized aggressive periodontitis at some sites the bone destruction does not proceeds further and it becomes stationary. Thus, it was proposed by Rane that the localized aggressive periodontitis is a self-limiting disease. The amount of bone destruction reaches a certain point and it stops. It does not proceed to other side. This phenomena is known as burnout phenomena. Now, before going into the microbiology of localized aggressive periodontitis, you must be thinking that why aggressive periodontitis is affecting the localized sites and not the whole oral cavity. So, why localized aggressive periodontitis involves molars and incisors? As you all know, molars and incisors are the first teeth to erupt into the oral cavity. They come at the age of around 6 to 7 years of age. At that time, the host's immune response is not that much strong in a child. So, when the aggregatibacter Actinomycetum comitans reaches these sites, the immune response of the body is not that strong that it can fight with the AA. So, AA produces various virulent factors which we have seen before, and it causes destruction at these sites. It causes bone loss and periodontal loss. Now, as AA progresses to the other sites, the age of the individual also increases and the immune response becomes more stronger to these bacteria. Thus, after the initial reaction, it is seen that the individual's body response, immune response increases to these bacteria, and thus the immune defenses, which are neutrophils, macrophages, are stimulated, and thus they produce immunoglobulins IgG and IgM against the bacteria, which causes phagocytosis of the bacteria. Phagocytosis of AA, and thus decrease in AA activity, and thus the spread of AA to the other sites is restricted in localized aggressive periodontitis. Therefore, in individuals with localized aggressive periodontitis, we see a strong serum antibody response to AA. A strong serum antibody response to AA is seen in individuals affected with localized aggressive periodontitis. Now, Let us come back to the microbiology of localized aggressive periodontitis. The main microbial organism responsible for causing localized aggressive periodontitis is AA, Aggregatibacter actinomycetum comitans. This AA is a gram-negative anaerobic rod. It is a gram-negative organism and it is anaerobic. It does not require oxygen for its survival. So, this is the main organism which is responsible for causing localized aggressive periodontitis. It is seen that the levels of AA decreases as the treatment progresses. As we treat the individuals affected by LAP, localized aggressive periodontitis, it is seen that the amount of AA bacteria decreases in that individual. The other organisms apart from AA responsible for causing localized aggressive periodontitis are Capnocytophaga species, Echinella corrodens, Prevotella intermedia, Campylobacter and gram-negative isolates such as Streptococci, Actinomyces and Peptostreptococci. Now, let us go through some Meritus prepared facts which we have already read, so let us revise them once. Aggressive periodontitis is caused by gram-negative anaerobic rods, facultative anaerobic rods. It occurs in individuals of less than 30 years of age. Gottlieb was the one who coined the term diffuse atrophy of alveolar bone in 1923, and cementopathia means cemental defects in 1927. Gottlieb termed aggressive periodontitis as deep cementopathia and hypothesized that it is the disease of eruption, and that cementum is the one which initiated a foreign body response. The World Workshop of Periodontics in 1966 coined the term periodontosis. Now, why periodontosis? Periodontosis means there is a destruction of bone, but bone inflammation is not seen. Destruction of the bone is seen. Destruction of bone is seen, but inflammation is absent at the involved sites. Inflammation is absent. Thus, the term periodontosis was coined. Now, Chaput and Butler in 1967 and 1969 coined the term juvenile periodontitis, which we have already seen. World Workshop of Clinical Periodontics in 1989 classified aggressive periodontitis into localized aggressive periodontitis and generalized juvenile periodontitis. Localized juvenile periodontitis and generalized juvenile periodontitis. World Workshop of Periodontology in 1999 classified aggressive periodontitis into localized aggressive periodontitis and generalized aggressive periodontitis. Previously, it was known as juvenile because it occurred in adolescent age group, but later it was found that it can be seen in older age group also. So, the term aggressive periodontitis, juvenile was replaced by aggressive. Now, A produces 100 kilodine protein. These are the outer membrane proteins, and these proteins are responsible for the leukotoxic activities of the AA. Now, TLR9, what is TLR9? TLR9 are the toll-like receptors. These toll-like receptors are present on AA, and these toll-like receptors are responsible for production of interleukin 8, increased production of interleukin 8 into the host, which is responsible for the bone destruction. Thus, these toll-like receptors, toll-like receptor 9, which is present into the AA, is responsible for the production of interleukin 8, which is a cytokine, and this interleukin 8 is responsible for bone resorption. Now, EvaluCite. What is EvaluCite? EvaluCite is a chair-side diagnostic test. It is an enzyme-based immunoassay This is EvalueCite is an enzyme based immunoassay. It detects various enzymes present into the bacteria and thus helps in recognition of the various bacteria like Porphyromonas gingivalis, Prevotella intermedia, Aggregatibacter, Actinomycetum comitans. Now what happens is, this is a chair-side test. So in the patients which are affected by these organisms, a subgingival plaque sample is collected and it is placed inside the kit. There is a box of EvalueCite on which a strip is there, and on that strip the plaque sample, the calculus and plaque sample are placed, and thus if these organisms are present into the plaque, it demonstrates a pink spot. Now let us discuss few questions of this topic. The first question is, first clinical sign of localized juvenile periodontitis. The word that you have to remember and you have to focus is clinical sign they have asked. As we all know that in localized juvenile periodontitis, first initial clinical features which are seen are tooth mobility and pathologic migration. Both are seen, but pathologic migration is a symptom which the patient will come to us with the complaint of gap between the teeth. Okay, so it is the symptom, but first clinical sign is a thing that we can make out, which the dentists with the doctors can tell ki okay, the tooth is mobile. Unless and until it is very much mobile, only then patient will come to know. But slight mobility is detected by the doctors itself. So the first clinical sign of localized juvenile periodontitis is, number 1, tooth mobility. Yes, this can be the answer. Gingival bleeding. Gingival bleeding is also a symptom which the patient notices. Pathologic migration, no. So these two are not the answers. And vertical bone loss along incisors and molars. This is also not the answer to this question. The answer to this question is tooth mobility. The first clinical sign of localized juvenile periodontitis is tooth mobility. The next question is, localized juvenile periodontitis is dash type of disease. As we already know that localized juvenile periodontitis is a degenerative disease because it causes degeneration of the bone, degeneration of the tissues and degeneration of the cementum. It does not, their inflammatory component is very less in localized juvenile periodontitis. It is present in generalized, but in localized it is very less. So localized juvenile periodontitis is dash type of disease. It is an atrophic disease? No. It is an inflammatory disease? No, because the local factors are very less. It is mostly because of the genetic defect and environmental interaction. It is not because of the inflammatory component. The inflammatory component of localized juvenile periodontitis is very less. So this is not the answer. It is a degenerative disease. Yes, it causes degeneration of the bone, degeneration of the cementum. Therefore, localized juvenile periodontitis is a degenerative disease. The answer to this question is number 3.